Welcome, everybody. I'm Greg Slobodkin, editor-in-chief of scienceboard.net, and I'm glad to have another interview with a KOL in the industry. We're with Brandon McNaughton, who is CEO and co-founder of Akadeum Life Sciences. Brandon, welcome.
It's nice to be here. Thanks for having me, Greg.
Absolutely, and so it's great to be talking with you. I've looked at your bio, and you've had quite the journey, as both an entrepreneur and a scientist. You've worked as an entrepreneur in residence, and also you've been a lecturer at the University of Michigan, and now CEO and co-founder of a biotech company. I guess my first question right off the bat is, what challenges are you looking to address at Akadeum Life Sciences?
How I think and what has allowed me to do what I'm doing now in this diverse background, like you mentioned, is really exposure to two things. Exposure to important technical problems, originally being trained as a scientist and how to solve those, but then getting more into, being a CEO, working with students, and those type of situations have really got me exposed to people. And when we look, at least for me, at what we can do as technologists, what we can do as even in the biotech industry, it's really to help people. So everything I've done, and I'd say everything the company is doing now, is really with an effort and a vision and a goal to help people. And that actually is across everything that I've done personally, but also everything that we do at the company. So for example, I even think science itself is really about us. It's about people. It's about us wanting to understand the world. It's about us being exposed to nature. And then, if we get into then companies, well, what's the purpose of a company? I think an ideal company is designed to help people, to make better products, to make people's jobs easier and better, and help everybody in their day-to-day activities. So that's one thing I've been exposed to, and also one thing I actually became an expert at and even teach. So the class I developed at the University of Michigan is actually on something called customer discovery. So this idea of human-centered design. And so we like to think here, we are a technology company, but we put people at the center. So the scientists, what they're facing, what tools they need, and making sure that we are helping provide tools and solutions to what they're doing. So that's everything that we think about is putting people first.
And it's interesting. I mean, when you look at your company's technologies, cell separation is clearly an important offering. Talk to me about, why cell separation is important and what are the different approaches to isolating cells?
Yeah, cell separation is important for a lot of reasons. I'll hit a few, but from the most kind of basic viewpoint, a scientist works right to understand the world around them. So if you're studying cells in the body, there's hundreds of different types of cells in the body. So if you want to say, understand the immune system, which is really a miraculous mechanism we have in our bodies, I think it's actually underappreciated. It's infinitely complex. It's just a wonderful thing. You have to first understand what you want to study. So say you want to study T cells and how T cells work. Well, you need to get those away from all the other cells. So that's the step of what we call cell separation. And if you think about, well, why is that significant? Well, understanding the function of the T cell then allows you to go and maybe do applications. So if we look at what's one of the most exciting things happening right now in medicine, it's cell therapy and CAR T therapy. Well, CAR T therapy needs T cells in order to be able to use them as a therapy, right? It's the T cell itself that you use as a therapy in CAR T. And if we never isolated those, we would never have understood them, but two, we could have never used those as a therapy. So that's just one example of how important separation is for the life sciences. And that's true in other areas, like just to pick one other one. You could look at diagnostics, and it's not even cell-based. Like if you wanted to, do a nucleic acid test to say, like COVID, right? How are a lot of the COVID tests done? Well, you have to isolate the RNA before you ever do a test on it. So that step of isolation and separation is really foundational to almost all biotech workflows. And it's something that, I like to think about it as like the unsung hero of the life sciences, because so many people are working on next generation detection technologies, which are great, but we also need next generation separation technologies. And that's what we're doing here at Akadeum. Your second question was about what are some of the ways that it's done now?
Exactly right. Yeah.
So this is a fun topic. I guess how I think about it as an analogy is you're going fishing, right? So if you think about like a blood vial, you have billions of things going on in there, all these different, cells and proteins and all these things. And the job of separation is to reach in there and pull one thing out. So Greg, if I gave you a vial of my blood and said, hey, can you get the T cells out of it? That's like giving you a bowl of a billion M&Ms and saying, can you get the blue ones out, please? So, that's the engineering challenge. So there's a few ways to do it. One of the most common ways, it's been around for about 40 years, is use tiny magnetic particles and attach those magnetic particles to, say, the T cells or the other cells, and then you can magnetically essentially pull them out. There's starting to be some other ways to do it. You can do like density. That's an old way of doing it, is use density gradients to try to it's almost like if you think about gold panning, it's the same idea. The heavy stuff goes to the bottom, the light stuff goes to the top. Imagine doing that with cells. So that's another way. There's some more emerging ways too now with using sound, and there's some efforts with microfluidics. But it's such a valuable thing that, I would say the community is really looking at lots of different ways right now to try to solve some of the foundational challenges with separation, which is again what we're doing here at Akadeum and why we think it's so important.
And so microbubbles, that's a technology that is near and dear to your company, and you see as a real solution in this space.
Yeah, we do. And that actually came out of years and years of being in the lab ourselves. So, if you ask a question, well, like why microbubbles and why now? So if you look at what's happened, is a lot of the existing technologies that have been around here for about 40 years or so, they haven't dramatically changed, and they're really good at, call it smaller volumes or a couple samples. But where we're going with like cell therapies and even other applications, everything's going to larger volumes. So you're not talking like, a small volume like this, you're talking, something more like my coffee cup and even bigger, because you're dealing with blood bags, and it's even getting bigger than that. And so what's happened is those are challenging samples, very, very difficult samples for other technologies to process. And I was using some of those in the lab myself when I was probably about 10 or 12 years ago. And I got very frustrated with what was out there. And in fact, what happened was we weren't able to do our studies because of the limitations of the separation process. And it was bad enough where we just started asking the questions, well, is there a better way? And why do we use it? Why do we use the technologies we do? And if we started over, how would we do it? And I think it was Edison said, if there's a better way, find it. So we found ourselves in that situation. And so we started brainstorming and said, if you just kind of started over with a clean sheet of paper, what's the simplest and most preferred way to separate out, kind of fish out those T cells or whatever you're going after in the sample? And we said it would be really nice to float what you want to the top, everything else sinks. And now that's where the microbubbles come in. And what we do is we essentially have these tiny floating particles, we call them microbubbles. They attach, say, to the T cell, and then that gets floated to the top and everything else sinks. And so that's how we do our process. And we like to think there's no simpler way of doing it, that this is really reduced to the best way of separating. And that means it's also scalable to those different volumes that I mentioned. Doesn't matter if it's a tiny droplet or a 10, 50 liter of fluid, the microbubbles just go up because they respond to gravity, not to something like a magnet. And then I have some here, Greg, if you'd like to see a demo.
Absolutely, yeah.
So yeah, I'll just show, 1 second here. I'll show this is actually a vial of a few million microbubbles. So that white layer on the top, that's after they floated up. So if you mix up the microbubbles, what you'll see, those microbubbles then can bind to the cells that are in the sample. And since gravity is active right now, those microbubbles over about 30 to 60 seconds will now float back to the top, and anything else in the sample sinks. So right now, in a matter of a minute, we're actually doing a separation together that would otherwise require gear, equipment, sometimes even instruments. So we simplify it so much that in some cases, you can eliminate the need for those instruments. And hopefully now, Greg, you can see the bottom there starting to clear up. That's because all the microbubbles have gone up to the top, where now they're recoverable, and the cells that you'd be looking for would be attached to those, and you can pull them out of the vial. And that's the technology.
That's really interesting. It makes me think, from an environmental perspective, right? The amount of toxic waste that the biotech industry produces has got to be extensive. I'm curious, are you able to quantify that? And how are separations currently traditionally performed in research?
It's a great question. And this is something like the amount of toxic waste just generally that is produced by being in the industry is significant. And if you go around the different labs and just talk to people, there is a real concern of, hey, we're working to do good, for other people and improve human health, but are we doing environmental harm in doing so? How much waste is generated, and what is the quantity and quality of that waste? Is it toxic? Is it recyclable? So this is something that's in the conscious mind of a lot of people right now. In some cases, there's actually efforts to move to all recyclable plastics that are used and disposed of in the life sciences. As far as separation itself, I think it's quite interesting because one of the most common ways to recover cells right now, and this is the technology I was mentioning, it's been around for about 40 years, is to use magnets. So you have to use very strong and what you call rare earth magnets to essentially pull those magnetic particles with cells out of a sample. And so what that means is they need, like the name says, rare earth metals. So if you just kind of step back and say, well, where does rare earth metals come from? Well, they require some pretty aggressive and damaging mining techniques and methods. So to give you an example, like just 1 ton of the material, the rare earths that go in the magnets, will actually leave a ton of toxic and even in some cases radioactive residues. So in China, for example, there's entire tailing ponds that are essentially toxic sludge. And, this is a real issue right now because the demand for rare earths continue to increase. But here with our technology, you don't need that magnet. So you completely get rid of the need for the magnet, which means now you can do your separation without that type of impact and damage to the environment. It completely goes away. And so I think about it as like, in some applications, like wind turbines, for example, have magnets. Well, there's not a replaceable thing we can put in the wind turbine instead of a magnet. But in separation, we literally don't need it anymore. Our technology has completely allowed for people to do separations without the need for a magnet. So why keep using something that's doing all this damage if we don't need to? And so that's one thing we're very excited about here at Akadeum is, it goes back to the helping people. We want to help people in their work, but we also don't want to do harm in the environment. So that's something we care about and think about, back to kind of our core ethos and culture here about putting people first.
Yeah, I mean, I think you answered my question as a follow-up, which was, how do your separation methods eliminate, the production of toxic waste on a scale that is currently ongoing?
Yeah, it's think about it as like, to kind of get some more numbers around there. There's tens of thousands of scientists around the world doing separations. And if all those people have magnets, that adds up. Because in those magnets, you're using some pretty exotic materials. So neodymium is one, which is like, I don't think I've heard that word except associated with a magnet. That's a rare earth. And so you get just so much pollution from just pulling out that material from the earth that we don't need to do. So like going back to like the demo I shared with you, you'll notice that like what was missing from that demo. There was no magnet. There's no piece of equipment. There's no microfluidic. It's just a tube and our reagent and our microbubbles inside of it. And because of that, it just gets rid of the need for all of those otherwise necessary pieces of equipment. And so that's basically how we get rid of it, is because we recover the cells with a different force. Instead of magnetism, we use flotation.
Very interesting. And so if we take a step back from the environmental and toxic waste issue, just generally, I mean, how does your technology improve the process, in areas like cell therapy manufacturing, clinical diagnostics, and cell and molecular research?
How our microbubbles improve the current workflows? There's a couple ways. And in fact, that's the reason we started the company. So again, it goes back to our own pain point. And, we went and talked to our colleagues too, who had similar pain points. But what does that look like? So just so I can give you 3 kind of quick examples. One is in the research space, like if you're looking at people who want to do sequencing. So single cell sequencing is really popular right now. Well, with single cell sequencing, you're trying to get an individual cell and then you run it and, you analyze the DNA. And so in order to do that, you have to have a lot of healthy cells. They have to be alive. They have to be viable. So right now, a lot of the samples that people have, they're low viability, meaning 20, 30, 40%. So they literally can't go run their sequencing because they have frozen samples that need to be essentially improved. And so that's one thing that we do, is we actually can pull out all the dead cells and leave behind the living cells so people can now go do their sequencing. Another example is with cell therapy. So if you look at where things are going with cell therapy, it's higher volume and higher throughput. So if you look, like there's, over 2,000 clinical trials going on right now with cell and gene therapy. So a lot of those require some sort of tool to separate that could be high throughput and high volume. So if we're talking about using, say, CAR T therapy to treat solid tumors, I mean, we're not talking hundreds of patients at a time. We're talking tens of thousands. So what is that scalable technology? Well, the existing ones really can't be scaled to that level. And so our technology allows, and is very kind of natural to go up in volume, because it doesn't matter if it's a tiny droplet or a huge volume. The microbubbles just feel gravity the same way and they go up. So we solve all those volume type problems. We have a lot of workflow problems we solve. And it turns out, because we don't use these really strong forces, these really strong magnetic forces, the cells are exposed to less force. So that means they're treated more gently. And if you're talking about cell therapy, one of the biggest problems right now is exhaustion. So, if you're doing all this engineering to a cell outside of the body, you have to be careful because you can exhaust the cells. Well, we've seen where our process is more gentle and can alleviate some of those pressing issues, in cell therapy. There's some diagnostic applications as well. But those are two big areas, at least, that we see our technology being the solution for, and that we currently are solving.
Wow, that's fascinating. I got to tell you, I will never look and think about microbubbles the same way. Appreciate your time today. It's been great talking with you, and we'll be watching Akadeum as things progress.
Thank you, Greg. Appreciate that, and thank you for having me.
Thanks, Brandon.